class, I have discussed about the introduction to the animal cell culture. I gave a brief introduction about that. So today we will discuss about preparation of culture media and different types of culture media. First of all, why you need to prepare the culture media? Media is required for the development of animal cells or organs or tissues, whatever it is. Okay. So for that we need synthetic medium. The medium may be either liquid or solid. So we will discuss in detail in the upcoming slides. First one, cell culture. Cell culture is the process by which cells grow under controlled conditions, generally outside of the natural environment. So tissues or cells or cell organs which are produced in a synthetic medium, which due to the compositions in the media. Media may be a natural media or artificial media. This is a general process for the preparation of culture cells. First, we have to harvest the cultures. The culture which has to be harvested only during the late log phase of the animal's cells. Okay, and when you have isolated or harvested the cells from the late log phase, then you have to isolate the cells with the use of appropriate enzymes, then which undergo the application, that means apply the isolated cells onto the appropriate growth media in culture birth. Then culture the cells by placing a culture dish in a cell incubator, which is shown in the figure. Next, you have to subculture it and verify the culture cells of the cell type, and cells are ready to be manipulated or modified for the experimental procedures. First one, animal cell culture. In vitro cultivation of animal organs, cells and tissues at defined temperature using an incubator and supplemented with a medium containing cell nutrients and growth factors, which is collectively known as animal tissue culture. This is what I have explained in yesterday's class. Okay, so we will go directly to the media composition. Media composition. See, even when you take preparation of plants or animals, that medium should be an essential one. So for medium which contains, in case of plants, you need carbon source, nitrogen source, surfactants and some trace elements. So in case of animals, we need for the development of growth, and it's very important, nutrients and growth factor. Growth factor, I already told you in earlier class, it is serum. Okay, so here similarly when for development of plants, here nitrogen sources is amino acids, energy sources, that is a carbon source, maybe a glucose, fructose or amino acids. And vitamins, generally we will add vitamin B and C. And inorganic salts, maybe a trace elements, sodium, potassium, calcium and magnesium. Okay, so next one, fat and fat soluble components, maybe a fat solid. And already I told you regarding that antibiotics, because which inhibit the growth of unwanted cells in the medium, that is antibiotic. Antibiotic may be a penicillin or streptomycin. Okay, then growth factors, that is obviously serum, then oxygen and carbon dioxide constant. Already CO2 inverters already told you, and inverted microscope to analyze or to view the cells which is grown on the medium. Then you have to go for the centrifuge for the cultivation of cells. For that we need centrifuge, then cryopreservation and serum. Next one, animal cell media, which is vary in their complexity, but most contains amino acids, vitamins, source and glucose, and corresponding concentrations are given in the slide. Next one, characteristics. This is already told you, primary cell culture, secondary culture is primary cell culture, anchorage dependent and adherences, and suspension culture, it is independent because it float on the liquid, so there is no need of attachments for the growth. Next one, finite and continuous cell lines. See, finite essence, it is a limited number of cells and it is a definite growth and it won't multiply after a particular time. So that is finite. In continuous, it is unlimited multiplication of cells, and most of the cells are finite cells, and some of them are only continuous. It divides infinitely, that is continuous. But in case of finite, which has only a finite number. So this is a representation of primary culture and secondary cultures and subculture and see, for example, you can take the tissues from the either animal or the plant. I will go for primary culture. From the primary culture, you have to subculturing. Subculturing, we go secondary subculturing, that is cell lines. Cell lines may be two types, is finite and continuous. Culture media. So here, culture media, it is there are two types. One is natural media and another is artificial media. Natural media, there are four types. One is biological extracts, clots, tissue extracts, and complex natural media. Similarly, in case of natural media, it is four types. It is serum-free media, serum-containing media, protein media, and chemically defined media. We will discuss one by one in upcoming slides. First one, the natural media. It is natural media, name itself, that any component or any factor which induce the growth of the animal cells which are isolated from the living systems. That is the natural. So it may be a coagulant or clot. Then biological fluids and tissue extract. First one, clot. Clot. The most commonly used clots are plasma clots. That is main important clot is used in the animal cell culture. Then in case of biological fluids, hemolymph, serum, and eye, insects, bloodstream cells, which are commonly used as a biological fluids. Next one, tissue extracts. Tissue extracts you can isolate it from you can see FBS, fetal bovine serum, and SBA, horse bovine serum, and bovine embryo extract, chick embryo extract. These are the tissue extracts which are commonly used as a natural media component for the growth of the animal cells. Next one, artificial media. Artificial media is entirely different from natural media. Here, the artificial media is used with in lesser concentration of natural. Maybe mostly they will use the serum. Since serum is in one media, they will have a serum. Another media which does not have the media serum. Okay. Another one, chemically defined media. In chemically defined media, there won't be any natural factors involved in growth of animal cells. Okay. So in case of artificial media, the advantages is prolonged survival, indefinite growth, and specialized functions. Suppose we need a particular vaccine. This, for example, polio vaccine, polio disease. We need to develop the cells accordingly for the particular functions. So, and immediate survival, that is prolonged growth, immediate survival. These are the main two advantages of artificial media. Next one, there I already told you the four different types of artificial media: serum containing media, serum free media, chemically defined media, and protein free media. In serum free media, that is first one, serum containing media. So we will take already told you, because whenever I say already told you, these are frequently used terms or important parameters which are dominantly used in animal cell culture. Okay, serum and fetal bovine serum, FBS, SBS, which contains almost 2 to 10%. And sometimes it has 10 to 20% also in the media. It provides basic nutrients for the cells, provides several hormones, contains several growth factors. The advantages of serum media is it is contained with growth factors and hormones and helps in the attachments of cells and act as a spreading factor. That is the main important advantage in the serum containing media. And another one is it is function as a binding protein. In case of disadvantages, lack of uniformity in the composition, and also increase in the risk of contamination. Contamination is another important factor which plays a major role. Okay. So next one, serum containing media. I already told you FBS is the most common supplement, and serum provides carriers, water soluble nutrients, hormones, and growth factors. Then protein free media, the name itself, it does not have serum. So serum is absent in serum free media. So but anyhow there is a few concentration present, since there are three things has to be noted because in this one there are three conditions. One is analytical approach, synthetic approach, and limiting factor approach. So the synthetic approach in sense that depends on the combination of composition added in the serum free media. An analytical approach, it is due to the analysis of serum concentrations, and limiting factor because for the initial survival we need serum. That is the limiting factor. Okay. Next one, chemical defined media. Chemically defined media, it is introduced in the year 2002 by James and Smith. So these two scientists developed the media which contains no serum at all and no any biological components present in the media. Only purest form of organic and inorganic constituents or compounds. Okay, it's contain because one compound is familiar polymer polyvinyl ester. This is a compound which is commonly used for production of animal cells in the CDM, that is chemically defined media. Okay, that is polymer alcohol. Okay, because this compound which supports the growth for the cells, whatever functions, whatever the support which is given by serum or other biological fluids, that same functions which are given by polymer polyvinyl alcohol in lesser extent or in small extent. Okay, next one, protein free media. Protein free media, it does not contain any protein at all, only and there is no biological fluids. Okay, so it is compared to serum supplement media. Use of protein free media promotes superior cell growth and protein expression of and facilitates downstream purification of any expressed product. Next, morphology of cells in culture. Cells can be divided into three categories: fibroblastic, epithelial, and lymphoblast. The corresponding pictorial representations are given. For example, in case of fibroblastic, cells are bipolar or multipolar in nature, which have elongated shells, which is shown in the figure. Next one, epithelial cells, which are polygonal in shape and more regular dimensions, and it is attached to a substrate, which is shown in the figure for the better understanding. Next one, lymphoblast. Lymphoblast is a like a cells which are spherical in shape and usually grow in a suspension media without attached to the surface. This is like suspension culture, which is shown in the figure. Next one, applications. Application is the production of antiviral vaccines, enzymes, cancer research, production of pharmaceutical drugs using cell lines, study of function of nerve cells, and it is used for recombinant DNA technology in genetic engineering. And this is, I'm going to conclude here that media, it is essential for the growth of animal cells, tissues, or organs. So, from this presentation, we should know, or we come to know that development of animal cells can be done either in natural media or artificial media. In natural media, there are four methods. One is biological method, that is biological fluids, clot method, tissue method, and another one is complex biological natural media. So, in that, some of the scientists like Dalmecos, Atkinson, Robinson, these three scientists involved in the development of natural media. So that is complex natural media. And in case of artificial media, there are four methods: serum containing media, serum free media, protein free media, and chemically defined media. These are the four medias which are used for the development of cells. So that is about the culture media in animal biotechnology, and in next class we will discuss about the organ culture development. Thank you for listening my presentations. Thank you.